Hello, everyone. A very warm welcome to my cytology classes. The topic for today's discussion is meiosis and its stages. Today we will discuss meiosis with special reference to plants. Okay, we are going to discuss meiosis that we normally study from the flower bud of Allium cepa. We'll stress on the accuracy of the diagrams, the number of chromosomes, how the outline should be. Okay. Now see, let's come to the topic meiosis in Allium cepa. Now, before we start, I hope you all have the idea about what is meiosis. I need not repeat that. All of you know what is meiosis, what happens over there, its reduction, division, all these concepts and ideas you already have, right? Here in this flower part of Allium cepa, we'll study two stages, two phases of meiosis, meiosis 1 and meiosis 2. Okay, meiosis 1 consists of separating the pairs of homologous chromosome. Now, if we write, see, meiosis 1. Okay, meiosis 1 is what? See, what did I say? Separating it consists of what? Consists of separating the pairs of homologous chromosomes, separating the pairs of homologous chromosomes, each made up of two sister chromatids into two cells, right? Each made up of two sister chromatids into two cells. That is daughter cells, right? One entire haploid content of chromosome is contained in each of the resulting daughter cell. Please remember, one entire haploid content, right? One entire haploid content of chromosome is contained in each of the resulting daughter cells. Right? Now see, the purpose of writing all these, despite telling, is that you also take it down in your notebook. The more you write, the better you learn. Okay. Okay. Now see, the first meiotic division therefore reduces the ploidy of the original cell by a factor of 2. From these two statements, what do we say? I'm just using a different color so that you remember the ploidy of the original cell. Ploidy of the original cell is reduced by a factor of 2. Okay. So this is what is happening. This is the ultimate result. All right. Okay. Now we'll move on to the different stages of prophase 1. Okay. The cells here are spherical to round in shape, spreading in nature. Okay. The nucleus present here is pretty large. You can see it. Chromatin fibers are visible. Okay. So see, these are the different characteristic features that you will start with. Okay. The first stage, you know, what is it called? You must know the name by now. You have studied this in your theory as well as you are studying it in your practical. You know what is the first stage? It is leptotene. Leptotene. Okay. You can see here, this is leptotene stage. Okay. So in leptotene, the chromatin fibers are visible. Correct? The chromatin fibers are visible. Look at this part especially. This is an original picture, not these. Look at this. So here you will see the fibers are very nicely visible. The fibers are nicely visible. Okay. Fibers are nicely visible. Okay. Individual chromosomes can be distinguished. The nucleolus is present and usually occupies a peripheral position. This is very important. The nucleus usually occupies a peripheral position. Nucleus usually occupies a peripheral position. Okay. So the nucleolus is nucleolus occupies a peripheral position. Remember this, right? So if you get to see these characters, you will identify this substage to be leptotene of meiotic prophase 1. So leptotene of meiotic prophase 1. All right. Next we come to the second substage called zygotine. Okay. Our next stage is zygotine. In zygotine, what do you see? Again, see the cells are spherical to round in shape, spreading in nature with prominent cell wall. These characters means what? It is pollen mother cell. Okay. These are typical characters of pollen mother cell. This will not tell you whether it is leptotene, zygotine. or any other stage. This is common to all, right? Next, what you should look for is the round shaped cell with large nucleus. Long, slender, paired chromosomes are present. Then you will look for this character. Long, slender, paired chromosome. What did I say? Long, slender, paired chromosomes are present. It will appear like this: long, slender, paired chromosomes are. You understand what I'm trying to say? Long, slender, paired chromosomes are present. Okay. Now see, homologous chromosomes now appears in pairs. Okay. A distinct nucleolus will also be here now. The nucleolus is quite distinct. See, if you again look back once to those photo, last slide, see nucleolus is visible here. Okay. So it is prominently visible. Hence, this substage is called zygotine. In zygotine, what will you actually see? How will you identify this? Homologous chromosome appears in pairs. Homologous chromosomes in pairs. All right. And they are long, slender, all those things you know. Correct? Okay. Next, what do we have? Next stage is Pachytene substage of prophase 1. So this one is zygotine. Zygotine. Next, we have Pachytene substage. Pachytene substage of prophase 1. How do we identify that? Again, you'll see this. This is Pachytene. How is it looking? Take a close look. That is why I'm going slow, so that you have no confusion. Identification is very, very important. You should know the key characteristic points by virtue of which you will identify the substage. See, sorry. Chromosomes are shorter and thicker than zygotine chromosome. In zygotine, what did I say? It will be long and slender. But here, it becomes shorter and thicker. It becomes shorter and thicker. Right? It becomes shorter and thicker. Okay. Shorter and thicker than the zygotine chromosomes. Homologous chromosome appears to remain coiled with each other. So these tend to remain coiled with each other. Okay, they tend to remain coiled with each other. These are thicker, these are shorter, and they remain coiled with each other. Okay. When you are drawing, try to make it look thicker than the zygotine chromosomes. Okay. Try to make it look thicker than the zygotine chromosomes. You understand? All right. A distinct nucleolus is also visible here. See, I'm only stressing on the drawing part of the work, okay? I'm not exactly describing each of the substages and explaining them. It has already been done in another video of mine. Please go to the cell biology, or as far as I can remember, it is there in my channel. There, the substages are explained in detail. Refer to that. For explanation, definition, whatever you say, refer to that. This is for making your concept clear. This video is solely made for making your concept clear. The rest explanations and everything, those are all in that particular video. Okay. So if you see these features, you will identify the substage to be pachytene. What is very important? Homologous chromosomes appear to remain. Chromosomes are shorter, thicker, and they appear to remain coiled with each other. Coiled with each other, right? Next, we move on to Diplotine stage. Okay. So what do we have in Diplotine? What are the characteristic feature here? The chromosomes are, it goes without saying, the chromosomes are short and thick. Okay. The paired chromosomes are distinct in shape. They're very distinct now. The paired chromosomes are distinct in shape. Several chiasma along the length of the bivalents are observed. This is the key point. Several chiasma along the bivalents. Along the bivalents are observed. Note this point. This is very, very important. The nucleolus diminishes in size and disappears during late stage. Okay. So if you see this feature, this particular feature especially, you will identify the stage as Diplotine substage of prophase 1. Now we'll draw. How is it looking? See. Look carefully. How am I drawing and how many am I drawing? Right at the beginning, what did I say? This is with respect to What? This is with respect to Allium cepa, where 2n is equal to how much? How many? 2n is equal to 16. Okay, this is how it actually looks. Is that clear? Is it? Ask yourself, ask questions so that your concept gets clear. So how many? 1, 2, 3, 4, 5? 1, 2, 3, 4, 5, 6, 7. 8. Okay. So if you see these things, you will call it Diplotine. Okay. Next, what do we have after Diplotine? It is diakinesis. In diakinesis, what will you see? The bivalents. The bivalents are distinct in shape. They're very short and thick. Some bivalents are lens shaped while some are scissor shaped. This is, see, this type of structure, they start forming here. But when you come to, or when the stage proceeds to diakinesis phase, that time it is becoming very, very clear. Nucleolus is totally absent. Okay, nucleolus is absolutely absent here. If we just write the key point, see, nucleolus is absent and bivalents are lens shaped, okay, or scissor shaped. Lens shaped or scissor shaped. Okay, understood? Terminalization is almost completed here. Bivalents are evenly distributed all over the pollen mother cell. See how I draw? Bivalents are evenly distributed all over the pollen mother cell. These two diagrams are very important. You should never go wrong here. You will never go wrong here if you know the concept, what is happening. If this is clear, no, you can never go wrong, never. And to get your concept clear, repeated study is required. One day before the exam won't help. Really, I'm telling you, one day before exam won't help. So 1, 2, 3, 4, 5, 6, 7, 8. Okay, this is how it will appear. Fine. Next, so this is which stage? Diakinesis. D-I-A kinesis. Okay, this is diakinesis. Next, we move on to Metaphase one. See, next we have Metaphase one, which will be followed by Anaphase one, Telophase one, Metaphase two, Anaphase two, Telophase two. Okay. So here we come to Metaphase one. All right. See, in Metaphase one, see, it appears like this. When you are taking the polar view, of course, you see, this is how it looks in polar view. It looks exactly like this. If you carefully observe, this looks very much like this. Okay. I'm again telling you, keep a note on the count of the chromosome. In one cell, 8, then 9, then 10. Please don't make this mistake. 1, 2, 3, 4, 5, 6, 7. One more, 8. This is Metaphase one. Metaphase one in polar view. This is Metaphase one in polar view. Okay. Here also you'll understand, see the bivalents are very short and thick, and they are arranged in equatorial plane in side view. See, if you see the side view, see, the side view is like this. I'll use a different color to draw the side view. See, if you see the side view, it will all be arranged like this. See, 8 chromosomes like this. This is side view. This is polar view, and this is side view. Okay, in polar view, the bivalents are well scattered in cell, like I have drawn here. Like exactly I have drawn here, it appears like this. But in side view, it appears like this. Okay. So this is Metaphase one. Next, coming to Anaphase one. I'm just erasing this. This is in polar view. Now coming to Anaphase one. How will Anaphase one look like? The chromosomes are seen in two distinct groups, and they move towards the opposite poles. Often, you know, you will not be able to count individual chromosomes in Anaphase one. So if you see that you're not being able to count the chromosomes in Anaphase one, please don't get confused. It is normal. You cannot count. Usually you cannot count chromosomes in Anaphase one. But just to make your concept clear, I'm drawing. In side view, it looks like this. Okay. In side view, it looks like this. Okay. Fine, it looks like this in side view. Next, we come to Telophase one. Next, we are going to study telophase 1. Telophase 1. Here, what will happen? See, the chromosome appear to form a lump here. You know the differences between early telophase and late telophase? I hope you know. In early telophase, you will not see the development of nuclear membrane. It is big. It is a lump-like structure. It is clumped, and once it becomes late telophase, the membrane starts developing. It will appear like this. Margin is uneven because the membrane is not here yet. Okay, nuclear membrane gradually reappears. Gradually. Okay, gradually reappears. Fine. And individual chromosomes cannot be identified. Right? This is telophase 1. Next, what do we have? Next, we move on to the meiotic 2 stages. So we'll start with prophase 2. Right? In prophase 2, what do we see? We see on both the sides of the cell plate, 2 nuclei are clearly visible. 2 nuclei are clearly visible. This is what we see. This is exactly what we see. 2 nuclei are clearly visible. This is 1. This is 1. Right? Individual chromosomes, as usual, cannot be distinguished. Prophase 2. Fine. And note that on both the sides of the cell plate, 2 nuclei are clearly visible. This is the key point. Right? Next, we come to metaphase 2. Okay. Next, we go to metaphase 2. In metaphase 2, what do we see? In metaphase 2, what are we seeing? The chromosomes are arranged in equatorial plane. In polar view, they appear in rosette form. So you see I'm just drawing this here like this so that you understand. When you are drawing it, please draw it neatly. It appears like this. In polar view. Okay. In polar view, it appears like this. Fine. Now, anaphase 2, same type of diagrams. I'm not drawing the borders any longer. You will see a structure like this. You will see a structure like this. Okay, just rough borders I'm drawing so that you just understand. Fine. 4 such groups of chromatids are present within the pollen mother cell. This is what you can see. Okay, this was what then? Metaphase 2, and this is anaphase 2. Clear? And last, what do we have? We have telophase 2. Telophase 2, I'll draw here itself. It's very easy, telophase, and very easy to identify as well. See, if I just make a cell out, make an outline like this, you will see chromatids are present in the poles. 4 such poles are present in the pollen mother cell, 2 each on both sides of the cell plate. If the cell plate is like this, see, this is more nicely formed. Like this and like this. This is your telophase 2. Okay. So this is something that you are going to see if you carefully study these stages under the microscope. Try to identify them by recalling the key characters that I have told you. Okay. So thank you so much for your patience. If you have any confusion, any doubts anywhere, please feel free to get back to me in the comment section. Till then, goodbye.